Bismillahirrahmanirrahim. Assalamu alaikum students. Today I will be discussing with you people HIV and AIDS. Let's start with our topic HIV and AIDS. Students, you know very well that HIV is an abbreviation which stands for Human Immunodeficiency Virus. Okay, HIV stands for Human Immunodeficiency Virus, and AIDS stands for Acquired Immune Deficiency Syndrome. Okay, Acquired Immunodeficiency Syndrome. One thing is indicative, students, in both these diseases, that both of these are disorders of the human immune system. So we can say that HIV and AIDS are viral infections which mainly affects the immune systems of the host. Now, the hallmark of or characteristics of HIV and AIDS. HIV and AIDS, the causative agent is HIV type 1 and 2 virus. These diseases are caused by HIV type 1 and type 2 virus, students. Number 2, both are disorders of the immune system. Both are disorders of the immune system. Number 3, they mainly affect cell-mediated immunity. Both these disorders, they affect cell-mediated immunity. How they affect cell-mediated immunity? They affect CD4 T lymphocytes. Okay, students? So these 4 points are the hallmarks of HIV and AIDS infection. That is, both of these are caused by HIV virus, type 1 and type 2. Both of these diseases, infections, they affect the immune system of the host. Number 3, they decrease cell-mediated immunity. And number 4, they mainly attack CD4 helper T cells. Okay? Now students, we move towards the details of these. Students, HIV belongs to RNA virus. RNA virus, which is a lentivirus family. Okay? Now, we move towards the transmission of HIV and AIDS. Transmission. Students, there are 3 main modes of transmission of HIV and AIDS. Number 1, sexual transmission. Okay, sexual transmission. Students, in this type of transmission, HIV virus type 1 and type 2, it spreads through male semen, vaginal and cervical secretions. HIV virus is present in these secretions mainly, although it may be present in other body secretions as saliva, as you can say in the sweat. So the Common route of transmission is sexual transmission through semen, vaginal and cervical secretions. These students. Second mode of transmission, parental transmission. Parental transmission. Students, parental transmission means through intravenous route, and this transmission mainly occurs through contaminated needles, non-disposable syringes, okay, IV drug abusers, parental transmission. And third, perinatal. Perinatal. Students, in perinatal transmission, we have three main stages. Number one, antepartum. Antepartum means before the process of childbirth. If the mother is HIV positive, then she may transmit the virus into the fetus. Number two, intrapartum. Intrapartum. Intrapartum means, student, during the process of childbirth. During the process of childbirth, instrumentation, use of non-sterilized instruments, use of infected instruments, other needles during the process of childbirth, they may transmit the HIV virus from mother to the fetus, as during cutting of the umbilical cord. Now, third one, postpartum. Students, postpartum. Postpartum means after childbirth. After childbirth, mother may transmit the HIV virus during breastfeeding of the neonate. Okay. Now, these were the three main routes of transmission of HIV virus, type 1 and type 2 both. Students, keep one thing in mind. Viral infections are caused by increased replicative rate of virus. Viruses multiply very rapidly in the host. And in case of HIV and AIDS, HIV virus must convert its RNA into DNA for rapid replication. This is the main characteristic point in the mechanism or pathophysiology of development of HIV and AIDS. I repeat my words. For rapid replication of HIV virus, this virus must convert its RNA into DNA. Now, how this conversion occurs? It is a sequence of process which makes this replication of virus more lethal for the host. Number one, attachment. Attachment. Students, when HIV virus enters the bloodstream, it becomes attached to the CD4 helper T cells. CD4 helper T cells. Students, this attachment requires chemokine. This attachment of HIV virus to the CD4 lymphocytes, it requires CD4 receptors, okay, which are located on the CD4 T lymphocytes. Number two, chemokine, receptors. They are present on the CD4 T lymphocytes. Okay, and special types of proteins, that is GP120 and GP40. Students, these two types of proteins, they are located on the envelope of the virus. CD4 receptors and chemokine receptors, they are located on the CD4 T lymphocytes. They have very high affinity for HIV virus, and these two envelope proteins located on the outermost surface of the HIV virus, that is protein 120 and protein 40. These four elements, they attaches the HIV virus type 1 and type 2 on the surface of CD4 T lymphocytes. Okay, students? Number 2 mechanism of it. Uncoating of virus. Uncoating of virus. Students, in this phase, when virus once has become attached to the surface of the CD4 lymphocytes, it becomes uncoated. The virus enters its contents into the CD4 T lymphocytes. Now, what are those important contents of the virus which enters the CD4 T lymphocytes? Integrase enzyme, reverse transcriptase enzyme. Okay, and number 3, protease enzyme. Students, these 3 enzymes are present inside the virus, and when virus attaches to the CD4 T lymphocytes, it enters these 3 enzymes into the host. Along with these 3 enzymes, viral RNA, which is in the form of 2 strands of single-stranded RNA. Okay, now these 4 elements are entered into the CD4 T lymphocytes. Now students, number 3rd step is DNA synthesis. Students, number 3rd point is DNA synthesis. In this step, the RNA of the virus is converted into the DNA under the effect of reverse transcriptase enzyme. Okay, the reverse transcriptase enzyme, it converts the single-stranded viral RNA into the double-stranded DNA. Okay, students. Number 4, integration. Students, in this stage, the viral RNA enters viral RNA which has been converted into the DNA. It enters the DNA of the CD4 T lymphocytes. Okay? In integration phase, the viral DNA enters into the DNA of CD4 T lymphocytes, and this action is influenced by integrase enzyme. Integrase enzyme. Here, transcriptase enzyme was acting in DNA synthesis. In integration, integrase enzyme is acting. And now, students, number 5th point, transcription. Students, in this phase, transcription phase, the double-stranded DNA of the virus, it becomes converted into the messenger RNA. You got my point? That in the stage of transcription, the double-stranded viral DNA, it gets converted into messenger RNA. It gets converted into messenger RNA. Now, students, number 6 point, translation. Translation. Students, in this phase, the ribosomal RNA, it forms proteins and enzymes from the messenger RNA. Okay? And these proteins and enzymes are called polyproteins. Polyproteins. In the stage of translation, the ribosomal RNA, it converts messenger RNA into formation of proteins and enzyme combination which are called polyproteins. Students, these polyproteins are the future viruses, and in the process of cleavage, which is the next step, cleavage. In this process of cleavage, these polyproteins, their larger fragments are converted into smaller fragments, and as a result, new viruses are formed. These smaller fragments of polyproteins which are cleaved are now termed as new HIV viruses. Okay, this was sequence of events which converts HIV RNA into the DNA, and this then leads to viral replication. Now, students, clinical signs and symptoms of acute infection, acute HIV viral infection. Students, the main clinical features are fever, weight loss, feeling of generalized weakness, which we call malaise, weight loss, rashes. We continue with clinical phases of HIV and AIDS. In acute phase, students, acute phase of HIV viral infection, viral replication load is very high. Clinical signs and symptoms I have already told you. Number 2, chronic asymptomatic phase. Chronic asymptomatic phase. Students, this phase of HIV viral infection, it has no clinical signs and symptoms at all. The immune system of the human host comes into action. The viral loads starts to decrease. But at the same time, there is gradual destruction of CD4 T lymphocytes. Number 3, students, number 3 is AIDS. When HIV virus destroys the cell-mediated immune system of the host, CD4 T lymphocytes are destroyed at a wider scale. There comes a stage when CD4 helper T cell count becomes less than 200 cells per microliter of blood. And this is the most terminal stage of HIV virus infection, which actually converts into the AIDS. And because of this decreased CD4 T lymphocyte count, immune system of the host is totally compromised. This HIV viral infection, it adopts the syndrome-like picture. It involves multi-systems of the body. It involves nervous system, respiratory system, gastrointestinal system. Almost all the systems of the body are influenced. And it is this stage when the infections which infects the host, they are termed as opportunistic infections. And these opportunistic infections, they are the characteristic hallmark of AIDS. So I wind up with these words. Inshallah, in our next lecture, we will continue our discussion on opportunistic infections in the AIDS syndrome. Thank you very much, students. Allah Hafiz. We discuss signs and symptoms of acute HIV viral infection. Students, HIV viral infection represents in clinical signs and symptoms of fever, feeling of generalized weakness, which we call malaise, lymphadenopathy, smaller lymph nodes all over the body, they become palpable and may be tender to touch. Number 4, rashes, weight loss. Okay. These and neurological signs, irritability, anxiety. These are the main signs and symptoms of acute viral infection. Students, clinically, HIV viral infection can be divided into 3 stages. Number 1, acute phase. Students, in acute phase, there is immense viral replication. The virus load is very high.